So today we are going to go over your Chapter 2 and 3 review packet in preparation for your test next Tuesday. So let's get that out and make sure that you have all the correct answers so you can study well for your next test. So let's start with what is an element. So an element is groups of similar atoms. Good examples of elements are the ones that we talked about as bulk elements, and that would be carbon. I'm going to write them out. Hydrogen, oxygen, and nitrogen. And bulk elements are elements that make up the majority of our body, 96% in fact. And so when I write little things like this on the side, you should also write that because all of this is necessary for studying well for your test. Trace elements. Trace elements are elements needed in small quantities in your body. And things that we talked about for that would be iron, we talked about chromium, we talked about potassium, we talked about sulfur and phosphorus. All of those would be examples of trace elements. The next question, number 4, asks you to go to a website to build an atom and to label all of its parts. So hopefully you did that because that's good practice and that gives you feedback as you build, but we can do that ourselves here. So we're going to build an atom of chlorine. I'm just going to draw a nucleus in here. And we know that chlorine has the atomic number 17. And so when we go to fill in the electrons for this atom of chlorine, we need to fill in 2 electrons in the first shell because that's how many the first shell can hold. We can fill in 8 in the second shell. That makes 10. And then we'll fill in the remaining 1, 2, 3, 4, 5, 6, 7 in the last shell. So orbitals are going to be these 1, 2, and 3. Okay, and it says to label the parts, so we're going to put 17 protons and 17 neutrons, and I'll label that protons, and I'm going to label neutrons. Remember, neutrons are neutral, they do not have a charge, so we're going to put that indicating the no charge, and then together the protons and neutrons are the nucleus. We'll label the electrons, they get a negative charge. And then it also says to label the valence orbital. And the valence orbital is the last orbital, the one that holds the outermost electrons. Alright, so we've labeled nucleus, we labeled proton, neutron, electron, orbitals 1 and 2. So this would be 1, this would be 2, and this would be valence. And we also labeled the valence down here. And now we're good. We have completely diagrammed chlorine, knowing its atomic number is the number of protons. So we start with that, 17 protons, and if I know 17 protons, I know we have to have 17 electrons. All right, moving on. So using the diagram that we just did, we need to answer these 3 questions. So the maximum number of electrons that the first orbital can hold, that is 2. Yes, it can hold less than that, but it can hold no more than 2. The maximum number of electrons in the second orbital, that would be 8. Again, it can hold less, but it can hold no more than 8. And the maximum number of electrons in the third is 8. Can hold less, but can hold no more than 8. So the octet rule states that atoms will gain or lose electrons to have a full valence shell. Because if atoms do not have a full valence shell, they are going to be looking to bond. So I'm going to add this over here. Full valence equals no bonds. Empty valence equals bonding. So bonding has everything to do with those electrons in that outermost level. If atoms have that level full, they're not going to make bonds. If they have space in that outermost level, they're going to be looking to either share electrons to make a covalent bond, or they're going to be gaining and losing to make an ionic bond. So valence electrons are really important when we talk about the behavior of atoms. So let's look at these couple pictures in number 7 and talk about the bonds that we see in each picture. So here we have water, so this is H2O, and in H2O we have an oxygen atom, which is the big one, bonding with 2 smaller hydrogen atoms. Oxygen has an atomic number of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Oh, sorry. It has a number of 8. And it's sharing this electron and this electron with these 2 hydrogen atoms. So because it's sharing electrons, this is going to be a covalent bond. And I'm going to put sharing electrons. Over here we have sodium, which has just 1 electron in its outermost orbital or valence shell, and it is donating or giving that electron to chlorine so that chlorine has a full valence shell, and then sodium would drop down to having this be its outermost shell. So here we have a gain or loss of electrons that creates charges, and then these charges attract one another. This is an ionic bond, and we have gaining / losing electrons. And then lastly, we have 2 water molecules here being attracted to one another. And this is an example of a hydrogen bond. And so which one of these is the strongest for number 8? So covalent is the strongest, and that is because something physical is being shared. So we actually have electrons being shared, and that's keeping them together. The weakest is going to be the hydrogen bond, and that's only because we're talking about partial charges or really weak charges attracting one another. All right, so moving on to number 9. So number 9, so what do we call atoms that have a charge because they have gained or lost electrons? So atoms that have gained or lost electrons are called ions, and ions then can be positive or can be negative, so they can be either one. I'm going to put positive or negative. But most atoms are not ions. Most atoms are what we call neutral, and they don't have a charge, and that's because they have equal numbers of protons and electrons. In other words, if you have a plus and you have a minus to cancel it out, you're not going to have an overall charge. So if atoms have the same number of pluses and minuses, they're not going to have any sort of overall charge. Suddenly, though, if they have one more plus than a minus, they're going to have an overall positive charge. Likewise, if they have one more minus than plus, they would have an overall negative charge. Okay, so now we'll talk about our 4 basic types of reactions. So our first basic type of reaction is depicted with A plus B equals AB. And this is what's called a synthesis reaction because we are building in our products. And if we turn that around and we have AB becoming A plus B, and this is breaking apart, this is a decomposition reaction. Decomposition. And then if we have AB creating A plus B, but it can go in both directions, this is what's called a reversible reaction. And lastly, if we have AB plus CD becoming AC plus BD, that's what's called a replacement reaction or double replacement reaction. You should have learned about those in chemistry. And then we're going to talk about polarity. So let's break this question into sections. So first we have what is a polar molecule? Second we have why is water a polar molecule? Third we have what is the effect? And last we have draw a picture. So let's start with what is a polar molecule. So a polar molecule is just a molecule that has a negative end and a positive end. A negative end and a positive end. Why is water polar? So let's talk about why. So water is polar because in the covalent bond that holds the atoms together, oxygen, let's just say electrons are shared unequally. The next asks what's the effect, and the effect is, and I'm going to write this down here and then erase it, the effect is that oxygen becomes negative and hydrogen becomes positive. And we can draw that as our little Mickey Mouse. So we have the two H's here, and we would have a big oxygen here, and we would have the negative down here, and we would have the positives up there. So then we had our picture. So when you do this question on the test, make sure that you're writing in complete sentences and that you're answering all those different pieces of that particular question. And then number 13 was straight off of your last quiz. What does pH stand for? And it actually has a bunch of different parts here too. So you want to make sure that here you also answer each of the different parts. So you have what does pH stand for, you have what is actually being measured, and then you have drawing the pH scale, labeling it, and including an example. So pH stands for the potential or power of hydrogen. What is actually being measured are the hydrogen ions. And then when we draw our scale, we have from 0 all the way up to 14, with 7 being smack dab in the middle. Anything below 7 here is going to be an acid. Anything above 7 up here is going to be a base. And if we wanted to include some examples, down here we might have lemon juice, or we might have even lower stomach acid. What we could put for neutral is pure water. And then a base, we could put ammonia, or we could put bleach. Most of the cleaning products we have are bases. So then we've answered every part of our question there. So if we add something to water and after we've added it we have more H+, that substance is going to be an acid. And if we add something to water and afterwards we have more OH, because remember when you split a water molecule you either get H pluses or you get OH minuses. If you have this, it's an acid. If you have this, it's a base. If you have equal amounts of H plus and OH, then that means it's neutral, just like pure water. All right, so let's move into our macromolecules. So the first macromolecule that we're going to talk about is our carbohydrates. So we're going to put carbs over here. Our monomer of carbohydrates are monosaccharides. And the elements that we find in carbohydrates are C for carbon, H for hydrogen, and O. And we always see them in a 1 to 2 to 1 ratio. In other words, we might have C6 H12O6, and that's an example of glucose. Functions, carbohydrates, we have three functions. First, we have that they are quick energy in the body. So glucose in our blood gives us that quick energy. Two, they store energy, and we store our glucose as glycogen in our body. And lastly, in some organisms, they provide some structure. So examples of carbs, so we already said glucose and glycogen. We have sucrose, fructose. And when we look at a carbohydrate, they look like a ring, like this. And they usually have things hanging off each end like this. So that would be a molecule of glucose. Okay, moving along. Next up, we have our, let's do proteins next. I'm just going to do them all in the same. Okay, so we have proteins. The monomer of proteins is amino acids. And here we have CHO, and now we have the addition of N. And we have 20 different amino acids for life. The functions of proteins are many. So we have movement because they help us with our muscles, immunity because they help us stay protected from illness and disease, structure, they help build things in our body, and then chemical reactions is a big one. They take care of our chemical reactions. And so some examples, so for movement we have the protein actin that helps us move. Immunity, we have things called antibodies. Structure, we could talk about the collagen that's in our face, or keratin, which is in our hair and our nails. And then chemical reactions are all done by enzymes. And so if we look at an enzyme, it's a big protein that has a pocket on the surface that we call the active site. And that active site fits a molecule that it's going to either help to build into additional molecules or break apart. So that's a good example of a protein. All right, next up we have our lipids. And lipids are made up of fatty acids. Don't put your lipids on my fatty acids. We have C, H, and O, but here we have more of the H and the O instead of the carbs, and we have no ratio for lipids. And we have three functions for lipids. The first is long-term energy storage. But then we also have cushioning and insulation in the body. Two examples, we have saturated fats and unsaturated fats. And remember that lipids kind of look like the letter E. So we have these fatty acid tails hooked up to this molecule that we call glycerol. And that kind of makes our lipid. All right, and then lastly we have nucleic acids. And our monomer there is a nucleotide. And our elements here are C, H, O, N, and P, chomp. And our functions are storage and transmission of hereditary information. So moving genes around. Examples, we have RNA and DNA. And a good example of a picture is if we drew our DNA here as a double helix. All right, so now we've covered our macromolecules. Let's get into a little bit more specifics. So it says compare and contrast saturated and unsaturated fats. So this is a really good question. So let's put saturated fats over here, and let's put unsaturated fats over here. So what we know about saturated fats is we know that they are solids at room temperature, whereas unsaturated fats are liquids at room temperature. We also know that saturated fats are unhealthier, whereas unsaturated fats are healthier for you. So the reason that saturated fats are solids at room temperature and that they are unhealthier is that their fatty acid tails have no carbon-to-carbon double bonds. So if I was looking at a big long tail of a saturated fat, all of my carbons are going to have a hydrogen attached. I have no double bonds between my hydrogens. And this means that those tails lie very straight. So fatty acid tails are straight and stack well. So when I put a bunch of these on top of one another, because they're so flat, they would stack really well. And that's what makes them accumulate in the walls of your arteries and helps to promote heart disease. So saturated fats you want to keep to a real minimum in your diet. Unsaturated fats, the fatty acid tails have these double bonds between their carbons. So we have C double bonded to C. And what happens there is we get these kinks in the chain. And that means that, and there would be hydrogens off of the ones that don't have the double bond, but that means that the tails do not lie flat. So tails have kinks. And they don't lie flat, and that means I can't really stack them very well because they have these weird shapes. And that means that they don't get compacted into your arterial walls and they move through more quickly and don't accumulate, so they don't promote heart disease like saturated fats do. So that's why we say that they are better for you than saturated fats. How are they alike? Well, they both have the elements C and H, and they both have these fatty acid tails. And you need both of them in your diet, but you just need saturated fats in far less amounts than you do your unsaturated. All right, so what molecule is considered fiber in the American diet? Well, that would be cellulose, which is a carbohydrate. And that is actually the walls of plant cells are cellulose. And so these plant cell walls don't get digested, and they scrub as they go through your digestive tract, and they help keep things moving, which is what makes fiber able to keep you regular, so to speak. And then the three basic components that all cells have are cell membrane, nucleus, and we mean an animal cell, do all cells, all animal cells, cell membrane, nucleus, and cytoplasm, that jelly-like substance that fills up cells. All right, moving on. So let's talk about each of the organelles and what each of the organelles does, and hopefully you've gone through the build a cell so that you see what it looks like again, because that's going to be important because you're going to have a labeling portion of your test with the cell parts. So the nucleus has a big important function, and that is storing DNA. And since DNA is the molecule that dictates how everything gets done in our bodies, the nucleus has a very important job of keeping this wonderful molecule safe. Structure-wise, it has a membrane, it has pores in the membrane that allow things in and out. So if we looked at it, it would have these little areas that would be kind of holes in the membrane. And inside it has another special organelle called the nucleolus. And the nucleolus is very important because it makes the ribosomes. And structure, we're going to say inside the nucleus. And if we were to draw both of these together, it would look something like this. So this darker spot would be our nucleolus. This would be our nucleus, and I'm going to draw the pores here in my nuclear membrane. And then inside I would have all the DNA, and some of it might actually be wound up into a chromosome. that you might see. It looks like an X. So it would look like something like that. The endoplasmic reticulum's very important job is to help transport protein through the cell. And we have two different types of ER. We have smooth ER and we have rough ER. Rough ER has ribosomes in it, and smooth ER has no ribosomes. And it's very important that the endoplasmic reticulum have ribosomes because in order to transport protein, something has to make the protein. So that's why it has those little ribosomes. Ribosomes make protein. So let's put that over here. That's a really important one. And so the ER is connected to the nucleus, and it looks kind of like this set of like little flattened sacs of membrane. And then the rough ER has these little dots on it that are the ribosomes, and then the smooth ER, which is further down, does not. Okay, so next we have the Golgi apparatus, and the Golgi apparatus acts a lot like a post office. And endoplasmic reticulum acts a lot like the interstate. And nucleus acts a lot like the brain. Okay, so the post office. So what does the Golgi apparatus do? So it tags, repackages, and delivers proteins. So it gets proteins from the ER. And so I'm going to draw it separate because it doesn't attach to anything out here. It looks like a lot like the ER, but it's not attached. So it gets the proteins from the ER in these little bubbles called vesicles, and it takes them in and it does what it needs to do inside, and then it ships them back out in a little bubble coming off the other side. And so again, it's just a flattened stack of membranes. And then often you'll see little bubbles around it. The mitochondria is a very important organelle. We call it the mighty mitochondria because it produces ATP, otherwise known as energy in cells. So super important, super important. And the mitochondria has two membranes because one of them does all of the energy creating, and the more of it, the more energy that gets created. So two membranes, and it kind of looks like a little oval shape, and it's got a second membrane here in the center that gets really folded up because, again, the more membrane we have, the more we have the ability to produce energy. And a cell may have 100 to 1,000 or more mitochondria depending on how much energy that cell needs. Your muscle cells would have thousands because those cells need that many. Lysosome sounds like Lysol, and lysosomes are the cleanup crews of the cell. They clean up dead cell parts and old cell parts and things that need to be digested, and they are little sacs of membrane with digestive enzymes inside. So they're little stomachs, really. They have the same stomach acid, basically, that your stomachs do. And when they fuse with food, then they can digest food inside of the cell. So basically, they're little stomachs for the cell. And they're very small, so they're about that big. And inside of them, they would have those digestive enzymes floating around, and they would be digesting things. And then lastly, we have the boundary, cell membrane. Cell membrane is important because it lets certain things in and out of cells. And it acts as a border for keeping all the cell stuff in and all the rest of the stuff out of the cell. And it has a very specific structure. So it's 2 layers of phospholipids. And then proteins, and then cholesterol. So if I was to draw the cell membrane, I would have these molecules that are the phospholipids, and I have one layer here, and then I would have the second layer turned in the opposite direction, and then inside I would have some molecules of proteins that have a space down through them that let things in and out. And then I would have little molecules of cholesterol stuck in here to help the cell membrane move. So phospholipids, proteins, the big things that let things in and out, and cholesterol. All right, we are cruising now. So let's just talk about active versus passive transport. So both of them move substances in and out of cells. But passive, which I'll put over here, uses no energy to do it. Uses the random motion of molecules. So because molecules move and bump around on their own, things like to go from areas where they are high in concentration to where they are low in concentration. So we're going down the slide here in passive. And then active, we are using energy, and we are going from areas of low concentration and pushing molecules into areas where they are already high in concentration. And so with active transport, we have a couple different types of active transport, just like we have a couple different types of passive transport. But the biggest difference between the two is the energy usage and the way that they move in terms of that concentration gradient. So here we would be, it's like you would have to climb back up a slide. You would be using energy and you would be going from low to high in active. So I like for you to think about that slide analogy, and if so that you can remember which direction they're moving in regards to that concentration gradient. So let's look at the next question, because that question will help us with the types of each of those. So osmosis is the movement of water, and water only, across a membrane. And that is going to be going from high to low, so this is going to be passive. Endocytosis is where we are using a vesicle, or that little bubble, to take in large things inside the cell. And that's going to be active, because the cell actually has to reach around and engulf whatever it's taking in that's large. Diffusion is where we're mixing two things based on random movement, so that's passive. Facilitated diffusion is just diffusion with help, and because it's still just a type of diffusion, it's also passive. Transcytosis, anything that ends in cytosis, it's going to be active. And filtration uses hydrostatic pressure, which is pressure and energy from the heart. So the cells itself aren't using any sort of energy, so this is going to be passive. So which type of transport moves large substances in and out of the cell by vesicles? So into and out of, that's going to be endo and exocytosis. Movement of water, we said, was osmosis. Pressure gradient, we just talked about pressure here, and that is filtration. Okay, so there are three different types of osmotic solutions, and those solutions do very different things to cells when cells are placed in those solutions. So I'm going to put a blood cell in each of these beakers. And my blood cell has all kinds of things in it, salts and sugars and other stuff. And so it has about a 5% concentration of those things, those solutes inside of my cell. So if I put a cell in a solution that was 25% salts and other things, that's going to be much higher than my cell. So this is going to be a hypertonic solution. And that means that water is going to go from where there's more to where there's less. So there's way more water inside my cell than substrate or solute. So in a hypertonic solution, water is going to flow out of my cell, and my cell is going to shrink. Here, if I have 5% and I put my cell into a 1% very, very fresh water solution here, there's way more water on the outside of my cell here. And this is what's called a hypotonic solution. And here, water's going to go from where there's more, which is here, to where there's less, which is inside of the cell. And my cell is going to swell and maybe even burst. Lastly, if I put my cell in a solution that is equal to it, it's called an isotonic solution, and that means that water is going to flow in and flow out pretty evenly, and the size of my cell is not going to change. All right, moving into cells. So we have 3 phases of the cell cycle. We have interphase, where a cell's doing its everyday job. We have mitosis, where the nucleus gets divided, and we have cytokinesis, where the actual cell gets divided. So mitosis, we just said, is the division of the nucleus. And we have 4 different parts to that. And we write it as PMAT: prophase, metaphase, anaphase, telophase. And here are the big things that you see happening in each of those stages. So prophase, we begin to see the chromosomes. So the DNA goes from a big plate of spaghetti to looking like our beautiful, cute little chromosomes in the shape of an X. In metaphase, the chromosomes line up in the middle. And in anaphase, the chromosomes are pulled apart. And lastly, telophase, the cell prepares to divide. And in that last step here of cytokinesis, it does divide. Okay, and lastly, what process allows cells to specialize and do different jobs? That is called differentiation. We are all born and we all have cells when we are just a few cells, and those cells are all the same. And what happens is those cells start to choose jobs. They become heart cells and brain cells and liver cells. And once they do that, that allows us to become more complex, because those cells start to form organs, and organs form organ systems, and that's what helps to build the organism. And the 3 reasons that we need the cell cycle are growth, repair. If you hurt your cells, you need to repair them. And reproduction. We have to make our sperm and our egg cells for reproduction. And lastly, if our cell cycle isn't controlled, what results is a tumor. Because if we can't control how the cell is dividing and our cells start to divide uncontrollably, that makes a tumor. A tumor is just a bunch of cells that shouldn't be there. And tumors can be cancerous or not cancerous, but sometimes they can be cancerous, and if they're cancerous, that means that they might spread and form tumors in other places of the body. All right, well, this concludes our Chapter 2 and 3 review. Remember, you can go back to this movie at any point in time over the weekend to study, and I hope you'll look over and do your best for your test on Tuesday. All right, see you next week.